Now we see today's new point, that is Mendel's experiment. Friends, we have already seen the terms of genetics, and we have also seen the definition of genetics, and we also seen some terms, terminology, and symbols that used in genetics. Now today we are showing the new point, that is Mendel's experiment. I introduce myself. I am Dr. G.B. Honna, Department of Botany, Sri Siddheshwar Mahavidyalay, Majalgaon. So, friends, the G.J. Mendel, the father of genetics, does an experiment on the garden pea plant, or it is also known as sweet pea. The botanical name is Pisum sataeum. Mendel selected the plant because pea plant are annually herbs and it is easy to cultivate. The flowers of the pea plant are naturally self-pollinated. The plant are homozygous, means the plant is pure. The flowers are larger in size. They are bisexual and they easy for emasculation process and also artificial pollination. The offsprings produced from the plants, these are by cross pollination and they are fertile. The plant show many contrasting characters. That's why the Mendel select the garden pea plant. Next point, the characters or traits selected in Pisum sataeum. There are two contrasting characters. One is dominant and another is recessive. In pea plant, the characters are, that is, height. The tall is the dominant character and the dwarf is the recessive character. Another character, that is the position of pods. The axial position of pod, it is dominant, and the terminal position, it is recessive. Then the growth of pod, in which there are two characters, one is the color of the pod and another is the form of the pod. The first, it is the color of the pod, that is yellow coat color of the pod, it is dominant, and the green coat color, it is recessive. The form of pod, that is fully developed, that is dominant character, and the constructed, it is the recessive character. The white color flower, it is the dominant character, and the violet color flower, it is the recessive character. The seed, that is cotyledon color and form of seed. The yellow color of cotyledon, it is the dominant character, and the green cotyledon color, it is the recessive character. Then the form of seed, that is round shape of seed, it is dominant, and the wrinkled shape of seed, it is the recessive. The Mendel select these 7 characters, and these 7 characters are alternate to each other, that is also known as traits, and these 7 traits are dominant, that is written in capital alphabetical, and those contrasting characters, that is recessive and written in a small letter. Then next point, the Mendel started his experiment on pea plant by considering one pair of contrasting character at a time. Example, tallness and darkness, in which some steps doing by Mendel. That first is selection of material, means the selected material, it is the plant that is selected 2 varieties. One is homozygous, that is tall, that is dominant, that's why written as capital T, and the other, it is also homozygous, but this is recessive, that's why it is written as small t. One plant is male and other plant is female. The next, that is the artificial crossing, that is known as pollination. Mendel collected pollen from male parent and it is dusted over receptive stigma of another plant, that is female plant. Mendel also used a tall plant and dwarf plant for performed reciprocal process. The offsprings obtained from the initial parents are called hybrids. That is, suppose he did the experiment capital T crossed to small t. From this capital T, it produces capital T gene, and another plant, that is small t, it produces, that is small t gene. Obviously, the formation of capital T and small t, they fertilize to each other and to form the capital T and small t, that is known as hybrid. It is also known as Inacean parents. According to Mendel, hybrids, that is capital T, small t, obtained through the parental cross are called as F1 generation, or it is also known as first filial generation. Selfing of F1 and F2, Mendel allowed F1 hybrids to self-pollinate to obtain next generation, that is the F2 generation. Then Mendel observed something. Mendel observed all F1 hybrids resemble with one parent and the character of the other parent was not seen in F1 hybrids. That is, tallness was appeared in F1 hybrids, but dwarfness was not appeared in F1 hybrid. Characters of both the parents, that is tallness and dwarfness, were reappeared in F2 generation. That's why the phenotypic ratio, it is observed, that is 3:1. And also he observed, that is F1 tall plant, the 1/3 were pure tall and 2/3 were hybrids and all dwarf were pure dwarf. And the conclusion of this Mendel experiment is that different characters in pea plants are controlled by genes. The genes come together in F1 offspring. They do not mix with each other, but they segregate at the time of gamete formation. See, this is the experiment, that is the capital T, small t, it produces the F1 generation, capital T, small t, it crossed selfly and to form the following generation, that is capital T, it is homozygous, tall, capital T, small t, it is heterozygous, it is also tall, and the small t, this is homozygous and recessive, it is dwarf. And the observation is that the phenotypic ratio is 3 tall and 1 bar, and the genotypic ratio is capital T, it is tall, capital T, it is also tall, it is 2, and small t, it is dwarf, it is 1. That's why the phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. Why the Mendel success in the experiment? Some secrets of this experiment. The first is selection of plant. He selected Physacium plant. He also shows the characters, accurate record, and last is Mendel luck. That's why this is the Mendel's work success to form the new type of experiment in the biotechnology. Thank you very much.